Now I'm going to talk about the seventh edition's chapter 7, which is talking about expression of lymphocyte receptor genes. So it's talking about how you can have such a wide variety of antibodies or T cells created within the body. Um, in fact, you have more antibodies in your lifetime than you have genes in your genome to encode them. So it was a big puzzle for a long period of time, and there were theories, but nobody could prove it for quite a bit of time. And then there's two researchers, um, Tsutsumi Tokunawa is one of them, and the other guy's name was Hosumi. Um, so Hosumi and Tokunawa uh, won Nobel Prizes and learned a lot about this process, and that's what they're famous for. Um, so, how is it that you can get so many different variations, especially in the variable regions of these antibodies, so that you have almost an infinite number of antibodies that can be produced within a population? That was the question. So, how can that occur? Um, and the analogy that I always like to use is, let's say that you have a chair factory, and in that chair factory, you're going to make as many different chairs as you can, but you can't keep extending the lines, the number of places that you work. So you do something kind of unique. You make five different kinds of backs, five different kinds of seats, and five different kinds of legs. Okay, so then when you start putting those together, you get 125 different combinations that could occur. Now, of course, some of those combinations wouldn't be pretty chairs, but you get the idea. By having multiple copies of the variable region genes and just a few copies of the constant region genes, you can increase the number of potential antibodies that you can produce through this process. Now, if you were to take that same chair factory and instead of just putting the back on, you used extensors or you cut a little bit extra off of the piece that you used as the back, then that gives you more variability in the way that you attach the backs, the seats, and the legs together. So by adding a few amino acids or taking a few amino acids away, you have increased your variability in the long run. But nobody really knew how genes could rearrange like this until Tokunawa started coming up with some of his research. So what we have here is the um, basically it's the recombination signal sequence. You're going to have to memorize this. The signal sequence is a combination of a heptamer and a nonomer. They're linked together with either a 12 or a 23 base pair sequence. Now, it's the 12 or 23 base pair sequence is always the same length. It's either 12 or 23, but the sequence in here is just junk. It's just spacer stuff. So the way this is organized is this is on the gene sequence, and they found multiple copies of these. And so you have an antigen receptor gene section that's connected with this triangular piece, which is the RSS, or the recombination signal sequence. Let's see, what else was I supposed to say about that? Okay, well, let's just look at some of these. This is a simple one, and this is basically what Tokunawa came up with. He found that there were multiple variable region genes, and there were just a few constant region genes. And then he also found some of these other genes in here that are called the J, which is the joiner regions. So if you are trying to make a light chain, remember the light chain has a variable region connected to a constant region, and it's a fairly short peptide. So you take any one of these three variable regions and join it with any one of these joiner regions, and then in the second round you would combine that with a constant region, and it could be one of a few constant regions. So by having different joiners and having different numbers of amino acids going between the V and J and between the J and the C, then you're going to increase the possible variable shapes that can occur in the variable region of just the light chain. Now, when you combine that with the variable region recombination that occurs in the heavy chain, then you have a mind-blowing number of possibilities. The whole idea for this started coming out when they were able to do some early sequencing experiments. And they were able to find that there were a bunch of different variable regions, but only a few of the light chain regions. And they also found that the same variable region could be connected to different constant regions. And that's just a simple basic concept that they were able to use. When they first started looking into this, they thought that there were two theories or two groups of people that thought that they had it figured out. One group thought that the antibody gene or the antibody receptor gene was in its entirety found in the genome. But they couldn't really explain why they had so few genes in comparison to the variability that you can get. Another group thought that there was a somatic hypermutation that was occurring. So you could get mutations that were occurring at a high rate, and that would cause an increased repertoire in the number of B lymphocytes. It's not exactly the case, but there is some hypermutation that can occur. Each generation of cells will produce slight variations in the variable regions that occur. And when they do that, they're increasing the likelihood or the possibility of one of the offspring being able to bind more efficiently to a specific antigen. So that's showing the same thing. All right, this shows Hosumi's and Tokunawa's experiment for DNA. So let's look at this for just a second. Anytime you're going through this book, pay attention to the experiments because they put them in there for a reason. So they found embryonic liver cells and they've isolated germ line DNA from it. So they found the DNA. And then over here they had antibody producing tumor cells of the same strain of mouse, and they were able to put the DNA over here. So they cut the DNA with restriction into nucleases and put it into gel electrophoresis. So if they blotted blotted it with a C region probe, they only found one binding site. DNA from the antibody producing cells, they found another one, but notice it's much further down on here. That means that this is larger. It represents a larger chunk of DNA. This is smaller. So something has been cleaved out of it to allow it to transfer through. The whole light chain, they found two, but they only found one over here. It's because only one is being expressed at this point in time. Okay, so that's basically what they did, is they were looking at the DNA in these B cells. They ran it out on agarose gels for 3 days at a really cold temperature because they wanted to get a lot of separation of the DNA. The gel was sliced and the DNA was eluted and hybridized with probes that bind to specific parts of it, of the DNA that's on there. And they were able to look for those regions that were called constant regions of the kappa light chain or the whole chain. And the plot shows the amount of radioactivity in each fraction as a function of the migration distance. It reflects the molecular weight of the DNA fragments. So they know that there was some post-transcriptional modifications that occurred in order to get some of those differences showing up. This is just a diagram that depicts the light chain versus the heavy chain and some of the differences in there. Remember, the heavy chain will be a much longer peptide when it's actually produced. So this gene has to have more constant region segments behind it. But it also has something different. I don't know if you remember from a couple of slides before. There's a V, a variable region segment. There's a J, which is a joining segment, and then there's a C segment that's downstream from there. And these three will come together, and it cuts out all this intermediate DNA that's in between these in order to bring it together. That's why when you run the DNA out in the previous slides, let's look at this. So this is the larger piece that's not started making into antibodies yet. And this is after it's been made into an antibody. So the VJC regions are connected here, and all that intermediate DNA has been cut out. So it's a much lighter molecule and it runs down faster. Now, the difference is here with the B heavy chain is you have VDJ in C. So VJC is in the light chain, VDJC is in the heavy chain. This slide depicts where the chromosome locations are of different genes encoding antibodies in both the human and the mouse. So you can see that the light chain genes are on chromosome 22 in the human and chromosome 16 in the mouse. That's the lambda light chain genes, and then the kappa light chains are on 2 versus 6. The heavy chain genes are found on chromosome 14 versus chromosome 12. So there are differences in the mice and humans, which means that some initial immunological studies can be done using mice, but you still have to do quite a bit of human trials anytime you're trying to develop a new vaccine or anything else, because there are differences in the way these genes react. Recently, and by recently I mean in the last few years, they've come up with transgenic mice and transgenic pigs and sheep that actually have a human immune system. So they've knocked out the animal immune system and they've replaced it with a human immune system so that now they can do final stage studies in those animals, and it would hopefully react much more like it would in the human being. Okay, so this is the organization of the immunoglobulin germline gene segments in the mouse. And let's see. Anytime you see this little psi symbol, that's a non-functional gene. It's there. It probably was functional in the past. See, there's 2 of them there. But now it's a non-functional gene. Some lethal mutation occurred. And if you ever have a recombination occur between the variable and the joining region here, then that would be a non-functional cell. And later in the semester, we'll talk about it. It would have to either switch over and use the maternal or paternal, whichever one it's not using, and try again to bring a variable region with a joining region. Or it would just be a dud cell and it would be cleaved out and killed off during the selective processes. So this is where it's talking about all the different kinds of variable and joining regions that can occur within the different types. There's the kappa and lambda, and then the heavy chains that you see. Many times in books it won't say heavy chain, but if you see the D sections in there, then you know it's a heavy chain operation that's going on. Table 7-2, I mean, there's not anything that you have to memorize from this, but just generally glance at it, and what you're going to notice is that there are a lot more variable region genes found in mice than there are in humans. Well, you have to think about it. They're living wild. They're constantly in battles and fighting and exposed to more antigens that may be out there. They're eating raw food that's uncooked in many cases. So we as humans have evolved to have fewer of these functional regions that are in there and have cleaned up the DNA. But the mouse immune system is actually much better at fighting off a lot of diseases that we are not. The Table 7-3 just shows a list of proteins that we're going to talk about individually in much more detail. There's RAG1 and 2, TDT. I don't know much about HMG 1 and 2 proteins, but I'll read up on it before I get to that slide. Cu70 and 80. Then there's Artemis, XRCC4, which has been really heavily studied recently due to its connection to certain viral diseases. DNA ligase 4 is a complex, and Cernose. It's named after one of the Greek myths. But we'll talk about those individually as we get to those slides. Where I'm going to introduce the concept of the one-turn, two-turn events that occur. So you're always going to have a 12 base and a 23 base that fold together. Usually you'll have a 12 base on, say, a joining region and the 23 base on the variable region. It's not always like that. It can be backwards. You can have the one turn over here and the two turn over there. The bottom line is that there's some spacer DNA in between all of these segments on a chromosome. And a lot of it is how it randomly folds over. And the one turn and two turn come together. It cleaves out the one turn and attaches the next piece of DNA right where that 23 base pair is occurring. So that's how it cleaves it in there. And so if you can imagine a piece of wire that you're twisting, and you're twisting the wire and it randomly touches between one of these one turns and two turns, and it melds together and becomes a loop. Well, the loop pinches off the excess DNA and it's gone. So that's basically how that works. So now let's look at the location in the germline. Did I go the right direction? Yeah, these are two conserved regions. And so you're going to see the one turn, two turn in different locations. And it's always going to be these two coming together. You won't ever get a two turn recombination occurring. So the genetics can be regulated by where they put the one turn or two turn spacer sequences that are in there. And that's kind of interesting in itself. So now let's look at this germline cluster that's going on. What we have is the number 3 is going to accidentally come together with the number 3 of the joiner region over here. So these fold over, and because it's a one turn, two turn, the spacer regions are cleaved out, and the 3 variable gene and the 3 joiner gene are recombined together in this next step of this recombined VJ gene inside the B cell. Then you can actually have, in this one it's talking about a heavy chain because you've got the D clusters. The D comes together with the J, so we have this segment come together with this segment and form the DJ rearrangement. And then that has to be rearranged with one of the variable regions that are there. So in the light chains, you have the V and J come together first. In the heavy chain, you get the D and J coming together first. This diagram demonstrates the activity of RAG1 and 2, which are all drawn together in this green circle that's on there. And basically, RAG1 and 2 causes the recombination to occur. It cleaves the nucleotides at the 1 and 2 when it folds over like this, and allows the recombination to occur. So it's going to cleave off this extra bit that's out there. The cleavage and processing of signal coding can occur there, and there's other genes that can come in, Artemis, TDT, DNA ligase 4, etc., that when they are involved in this, they are going to either add a nucleotide or take a nucleotide or two away. They're adding or subtracting codons that code for amino acids. So this diagram is much more complete, and I don't think I need to sit here and read it to you. You are all adults and college students, so you can read this. It's figure 7-8, and it's a mechanism of VDJ recombination that can occur. And it talks about how the different enzymes are going to jump in there and work on these processes. Okay, there are 5 major mechanisms that generate antibody diversity in naive B cells. Uh, and I want you to memorize these. This is going to be a question on the test. Learn them, understand them. Number 1, you have multiple gene segments. So we've already covered that. You've got multiple genes for the variable regions, the D and the J regions in the heavy chains, and the V and J regions in the light chains. You have just a few of the constant regions. But by having these multiple gene segments, you get greater diversity of possible receptors being produced. Number 2, you have what's called the P nucleotide addition, and that's when DNA hairpins, um, where the DNA pieces are being cleaved and brought together, reannealed, they result from cleavage that's not symmetrical. In other words, sometimes there'll be an extra nucleotide or two in one side of it, and they have to fill in the gaps. So that means one time, if you were to do this same combination of genes, you'll get 10 nucleotides in between, and the next time you may only have 3. So that can make a big difference in how those regions are joined together. So P nucleotide addition, you're adding nucleotides. Exonuclease trimming. There are lots of enzymes involved in this that you've been reading about. And what they do is they trim pieces off as they're joining the VJ sections or the VDJ sections. So it causes loss of nucleotides. So we have either the addition of nucleotides in one step or exonuclease trimming in another step. Number 4 is non-template in nucleotide addition. Okay, so the way this works is, especially the enzyme TDT, has several mechanisms that give rise to extra diversity at the junctions between the gene segments. In addition to these 4 mechanisms for antibody diversity, you can also have individual variation in the gene sections, the gene pieces. You can have recombination that occurs during crossover events during meiosis that may show up within a population every once in a while. And then, of course, last but not least, there's combinatorial diversity, and that's the same heavy chain can combine with different light chains and vice versa. Okay, before I talk about this slide, I want to give you kind of a quick definition of what affinity maturation is. Affinity maturation occurs in B cells around the variable region loci. And what happens is every time you get exposed to an antigen, the best memory cells or the best cells able to bind to that particular antigen or immunogen will bind, and then it will stimulate them to go through a clonal uh, expansion process where they make multiple copies of themselves. Each of those copies can go through a mutation that occurs in those very important variable regions. The next time you get exposed to the same antigen, only the best ones of that group get selected. So every time you get exposed to an antigen, you're going to go through what's known as affinity maturation, which means the first generation antibody to be able to bind to an antigen may only have a bare grip on it. But by the time you go through multiple generations of exposure to an antigen, then you start getting a antibody that's the main one to be produced that has a really good high affinity for the particular antigen that you're working with. So that's affinity maturation. Okay, because you have a mom and a dad, I'm going to give you some basic information here. Because you have a mom and a dad, you get one chromosome that codes for your light chain from mom and one chromosome or gene section from your dad that codes for the same type of light chain, but they're different because they're obviously different individuals. Same thing's true for the heavy chain. Well, this could be a problem when you're trying to assemble heavy chains and light chains together. So there's a process known as allelic exclusion that means that the first allele to produce a functional antibody, light chain or heavy chain, will be the one that's expressed in that cell line. Ooh. So wait a minute, is that not another level of diversity? Because you have some cells that will select for the maternal genes for the light chains and the maternal genes for the heavy chains. But then you can have other cells that have the maternal light and the paternal heavy, or vice versa. So you're increasing your diversity of types of antibodies that can be produced. And that selection of allelic exclusion occurs at the cellular level. So, the immunoglobulin gene, this is a pre-B cell receptor. And remember, we call an antibody a B cell receptor until it has been activated and starts being excreted as a soluble antibody. This is found on the cell surface with a variable region that's a pre-B and a lambda 5 region that's found on there. And that's it. This is just kind of, to me, it's a placeholder. It's like putting a piece of cardboard on there until you're ready to put the actual thing in place. So let's look at how you generate functional immunoglobulin receptors through this rearrangement complex that can occur. Heavy chains rearrange first. If something happens and the cell is unable to produce a functional heavy chain, then that cell is killed off. So there's a lot of apoptosis that can go into this. So we have a progenitor B cell, and it's going through heavy chain rearrangement. You know it's heavy because there's a D there. And it's becoming a naive B cell. That's the process. So it's here. Now, it can have an allele 1 or an allele 2. If it produces a productive allele 1, then it never goes back and does anything with the allele 2. So if this is the maternal, then it stops there. If that doesn't work, then it will go to a secondary allele. Yeah, so this is how it goes. Here's the kind of the pluripotent B cell. It's a stem cell. It has the ability to express either of the heavy chains. If it expresses one and it's productive, then it won't express the other one. So it shuts off that process. And then it can move on to the light chain events that occur over here. If it happens to produce a non-productive allele 1, then it has the chance at producing the other allele, the paternal or whatever allele it is, and producing a productive cell. If both alleles fail, you get non-productive alleles in both cases, then that cell goes through apoptosis and dies. So it's a selective process. The next step is to go into the light chain rearrangement. So you get productive allele 1 and it stops. Then you've got an IgM, monomeric form of the IgM on the surface of this virgin B cell. Where if it's non-productive, then it has another shot at it. It can produce an allele for 2, and if that's the case, then you have this antibody formed. If this one's non-productive, then it shuts down. And because you have different kinds of lambda and mu and kappa light chains, you actually have 4 shots at it to get through the light chain rearrangement. And that would produce a functional antibody. And you can see that this cell can produce multiple copies of this one, and it has the ability to express any of those alleles. And it's just really luck of the draw which ones get put it out there. So that increases the amount of variability that you can have. One of my favorite questions to ask on one of these exams is about class switch. Class switch recombination is a special event that occurs in cells after they're activated by their antigen. It results in a loss of DNA encoding the small segments down near the end of the FC region, and it can allow cells to switch from one class of antibody to another. So it's possible for you to have an IgM, IgG, an IgE, and even an IgA, all with the same exact Fab region binding site, the same idiotype. And that's because of this class switch. So I want you to be able to discuss class switch and how it works in immune cells. I would read through the rest of this chapter. It's a little complex, but the main things that I've talked about in this video are going to be the things that I will ask about on the exam.